Hello and welcome to BIS103, Bioenergetics and Metabolism for the spring quarter 2020. Unfortunately, given our current circumstances, this will be the remote edition of BIS103. So this will be the first of 18 lectures that I have pre-recorded and that you will have access to throughout the quarter alongside the slides, any notes that I may be taking during the lectures. And I hope, even though we cannot be together more interactively and learn biochemistry, that all those resources will be sufficient and work for everyone to master biochemistry this quarter. So let's get us started. Here we go. Slight technical glitch. So this will be section 003 for this quarter. So if you are enrolled in the sections 1 or 2, please make sure you are enrolled in the right one and let me know right away if there are any issues. My name is Philip Zerbe. I'll be the instructor for the spring quarter. And so what we will do today is just go through some course logistics, the lecture schedule, get you prepped for the quarter so you know what's coming, and then we'll start to dive into metabolism. So again, I said my name is Philip Zerbe. I'm an associate professor in the Department of Plant Biology. And so if you need to contact me with any question, with any issues, etc., my phone number is listed here, as well as my email. So you can contact me through this anytime, as well as through the Canvas chat. And just so you know who you're talking to and who's your instructor this year, my lab really focuses on plant biochemistry. We are particularly interested in plant natural products, how they are formed in plants, what their function is in plants, as well as how we can use them as bioproducts such as medicines, flavors, fragrances, biofuels, etc. So we're doing a lot of protein biochemistry, function genomics type of work, but also pathway engineering for the production of these natural products. To start with some logistics today, please make sure that you're aware of the prerequisites for this class. Specifically, this is BIS102. So if you have not completed BIS102 or you're uncertain, please contact me right away so that we can address that. BIS102 really is an important prerequisite for this class because it provides a lot of sort of foundational understanding of several topics that will be important for working through some of the topics we'll be discussing throughout the quarter. And so specifically, knowledge of amino acids that you will have covered will be really important. Enzymes and their reaction, we will look at a lot of enzymes, their relevant reactions within pathways together. And likewise, you should sort of be digging back into your organic chemistry classes too. We will be seeing quite a bit of functional groups that we'll be dealing with, different kinds of organic chemistry reactions. So oxidation, reduction reactions I have listed here as one important aspect. So if you feel a bit shaky on organic chemistry, it's probably a good time sort of ahead of the quarter and now to start and refresh your knowledge of that, but I will also be going through some of the basic foundational knowledge that we need for this course that relates to organic chemistry. Some of the general course information I have uploaded on Canvas in an info sheet that you will find there, and you can access there and download. Our lectures should have been Tuesdays and Thursdays, 12 to 1:30. Now they're pre-recorded. If there's an option that we can go live, for example, via Zoom, I will let class know if that possibility should arise. Other than that, you should expect to just work with these pre-recorded lectures for the quarter. How to get you can touch with us. So there are multiple options. We will obviously have office hours. You can reach me via email, and please, just for organizational ease, if you could put BIS103 in the subject line, then it helps me to organize the emails and get back to everyone as quickly as possible. And I also encourage you, especially this year, as we have to do remote learning, to use a Canvas chat, because that allows me to more efficiently get answers back to the entire class. Here again are my contact information. So in case you want to reach me, your TA for this year is Zerbe, and he will also have his own office hours. So you have two people that you can reach out to. In addition, we have an undergrad tutor this year, Libra McKee. He will be helping also with monitoring the Canvas chat room to try and help and address questions that you have throughout the quarter as efficiently and as best as we can. You see the times for the office hours here as they are listed. This may have changed by the time we go live for the spring quarter, so please watch out for announcements for office hours. We'll try to potentially extend them so that we have opportunity to address questions live and improve learning. Right? So please watch out for any possible changes that may have occurred after I've recorded this lecture. The book, I'm working with generally the Leninger, the Principles of Biochemistry. It is not obligatory for this class, but I think it's very helpful, especially this year for remote learning, if you can get access to the book. You do not need to purchase or lend the most recent edition. The 5th, 6th, or 7th edition will be fine. There aren't too many changes, and I have assigned some of the sort of key pages and key chapters of the book across these different editions, also in the lecture schedules, so that you have a little bit more ease for moving through the material. Okay? And you should be able to get this either remotely or, if still possible, by the beginning of the spring quarter, also through the library. I have ordered some books there for class. Exams, important information. We will have 2 midterms and 1 final exam. I have listed here the dates for these. So our first midterm will be on April 21st during our official class time. The second one, same time, a different day, on May 14th. And then our final will be June 8th here from 1 to 3 p.m. Please ignore the room numbers here. All exams, due to our current circumstances, will be online, and I will provide them as Canvas quizzes. Each exam will have 100 points each. Please note that the final exam will be cumulative. This is important in biochemistry because throughout the quarter, what we will learn will build on what we have learned before. And so it's important to have a cumulative exam at the end. But as you can see here, I typically ask about 60 to 70% questions about the more recent material you will have watched. And the older material from the beginning of the quarter will be asked in more general terms. In addition, this year, to sort of offset the stress of the 3 exams, I have also 3 graded Canvas quizzes that are worth 6 points each, and that I will provide periodically throughout the quarter, basically after each week, where we don't have a midterm or final after that week. You will have access to a quiz that is an open book at home, small exam. Okay. So I hope this will work for everyone. The lecture schedule, you can see here. Again, please note if there are any announcement for changes due to sort of the really dynamic situation and as things develop on campus. The current state, what I would walk through this quarter, is listed here in the lecture schedule. And so you can see here the dates. Now they're pre-recorded, but they're associated with the different topics here of each lecture, so you can be prepared for what you can expect to come. Then I have here listed the pages of the book relevant to these topics for each of the edition of the book that I would recommend. And there's some additional notes here too, what kind of chapters and what you should be focusing on if you want to do this additional reading and learning from the book alongside our lecture material. So what we'll do is, today we're here, we'll just start with a brief introduction on what we will be doing. After that, we'll start to look into our understanding of metabolic flux and metabolic pathways. We'll then start to look at our sugars, our carbohydrates, look at how these are broken down, used for energy through glycolysis. We'll be looking at some other sugars outside of glucose here too. And then we move deeper into energy metabolism. We were looking at the pyruvate dehydrogenase reaction and the TCA cycle, very critical reactions for energy metabolism. Then we'll have our first midterm here after this material. And after that midterm, we'll start to move into how we can actually make sugars through gluconeogenesis. We'll understand the pentose phosphate pathway and how it relates to metabolism more generally. Then we'll move into fats. So we'll look into how we can break down and produce fats as another energy resource. We'll move into the electron transfer chain and the production of ATP, so one of really key pathways for the production of energy-rich compounds. And so this material then will make up largely the material for our second midterm. And then moving on, we'll start sort of a first lecture on actually putting it all together. So taking the knowledge we have gained from these first two midterm periods to try and understand how are all these pathways working with each other interconnectedly and how they're regulated. And once we have sort of starting to have an overview here, we'll go in through our next big chapter, and that's proteins. So we'll be looking at amino acid reactions here. Again, similar to our sugars and fats, we will mostly be looking at how they're being broken down for energy and how they're being actually synthesized for different functions in metabolism. And after that, we'll have another lecture here, sort of taking all this knowledge together, looking at how this all can be integrated, really with focus on mammals and humans in this particular case. And at the very end, we go back to the beginning and look at where all this energy actually comes from. And we're looking at photosynthesis and how that works in just one lecture, right? And then we'll finish off with our final exam, again on June 8th here. So far from 1 to 3 p.m. And again, ignore this in the classroom. It will be a remote final as of now, unless there are any changes during the spring quarter. And after that, I'll send you off, and hopefully the situation will have improved and you can have a good, relaxing summer vacation, right? So let's dive in. Some of the learning objectives. So I have prepared learning objectives for each lecture, and you can find them with the lecture slides for each lecture, and you can download them on Canvas. They will be uploaded by the start of the spring quarter. And these are really designed to give you an idea of what you should expect, of what you should be understanding throughout the course. And so if you feel comfortable that you understand the materials associated with these learning goals, you should be perfectly fine for the midterms and the final exam, as well as the quizzes. All right, here we go. So this is from the syllabus, so just to give you sort of a broad overview of the learning goals. Also, these are now already available on Canvas. So there are a few things here that you can go through in your own time that really broadly speak to what we want to cover this quarter. And I'll be going through these learning goals sort of lecture by lecture in more detail to give you some overview. Some advice to sort of really help you succeed in this class. I really highly recommend that you watch these recorded lectures. If the possibility arises that we can do them live, I will likely do so to improve more interactive learning. but really pay attention to the class material. I would encourage you to read the text before and after lecture to deepen the knowledge, use all the recorded material again, and really give yourself time. BIS103 is one of the more advanced classes. It tends to really be one of the, maybe not harder classes, but it's a lot of material and it's not easy. So please give yourself enough time to study the material. There will be some structures that you will have to memorize, but not all. And I will give specific sort of guidelines and advice on which structures you really should be memorizing and which ones you can focus less on. So, right. Again, what I would really encourage you to do so is to try and go beyond memorization. Try and understand the chemical logic behind these reactions. And I will try and really explain to you throughout the quarter, throughout the pathways we will be discussing, on the underlying chemical logic of these interactions and the conversion of different chemicals. Because the more you understand the chemical logic behind these reactions, the easier it will be to not have to memorize these structures, but to be able to reconstruct them. And it will be really then knowledge that you can carry forward for a much longer time than simply memorizing. I think it will greatly help you. So I strongly encourage you to try and try to look beyond the structure and look at how these reactions actually work in their principles. And with all that said, please talk to me if you have any questions. Reach out to me at any time during the quarter. So myself, your TA, and our undergrad tutor are there to help you, especially this quarter, as I'm sure during all the remote learning, this will be not the easiest quarter. But I think we can do this together. So here's to that. We can do this. All right. More to study aids, again, office hours, there will be at least 2 per week. I am working on this actively and will update everyone by the beginning of the quarter or shortly before that on what the situation will be. Pay attention to your learning goals. I have updated a number of practice questions and practice exams to Canvas, so you can use these for your own learning. We will also cover some of these practice tests in our office hours. Please note, though, that they are slightly different from the exam that we'll have this year, simply because it will be a remote exam. The questions will be largely the same, but you can expect that they are likely less essay type questions and more multiple choice, true or false type of questions, just due to moving everything remotely. And again, please use the Canvas chat as you can. It's a good means for us to stay in touch and be sure that all the information really is translated to the entire class. All right, so this is what I have for logistics. Again, please don't shy away from contacting me. I would strongly encourage you to use the chat. It will be very good because it's an easy way for me to provide the information to the entire class. But by all means, if your question is sort of more of a personal nature and you don't feel comfortable sharing the question, please email me, contact me directly so that we can address it. All right, let's dive into metabolism. This is the fun part. So what we want to really do this quarter is to really understand the critically important and interconnected role of biomolecules in living systems. That's a mouthful, but what I really want to stress to you this quarter is how small molecules, metabolites, in general, biomolecules are really critical for sustaining life. And sustaining life all the way from sort of the origin of life and small entities of biochemistry to the evolution and development of really simple life forms such as amoeba here, to more complex life forms like us, and how biochemistry is really critically underlying the development and the sustainability of life, and also how we can use biochemistry to generate the energy that we need to stay alive, to move around, and to just be the way we are. And so there are multiple aspects to this. Really, the way you can see this is that metabolism is a science of change. Metabolism is the way in nature how chemicals are interchanged between each other to generate life, to maintain life. And so this is really the basis of what we want to go into. We will look at very closely at some of the shared and unique properties of metabolism. You'll be surprised how many foundational biochemical pathways, biochemical principles there are between microbes, between plants, as well as between humans. A lot of these components are very conserved and are really shared amongst all of us, all of life. But there are also some unique properties that different organisms have developed, and we will look at those too. And sort of in association with that, we'll also look at some of the old and some of the new tools that are available to us to study metabolism. How can we use research and science to understand metabolism? And sort of around that, we'll look at some specific examples. We'll look very deeply into the biochemistry of why you get a hangover. What's the biochemistry of that? We'll look into how our nutrition impacts our metabolism and vice versa. And we'll look at some sort of specific examples, how biochemical knowledge and biochemistry can be utilized to develop, for example, drugs for flavors, fragrances, other bioproducts that are really important for our daily life. And we'll try to explore some examples on how biochemistry really has led to some of these discoveries and developments. Right? So it should be a fun quarter. So let's start with some sort of basic terminology and definitions so that we get all on the same page of what metabolism, biochemistry, and bioenergetics really means to us and what we will be encountering this quarter. Right? So let's start off up here with bioenergetics. What is bioenergetics? So if you loosely define it, bioenergetics really is a study of energy conversion in living systems. So a big part of what we want to do this quarter is to try and understand the flux of energy. And flux of energy here means the production and the utilization of bioenergy. And so that utilization and production, then how does it relate to biochemical work? Biochemical work meaning, for example, us moving around. Right? For us to walk around requires bioenergy and it requires biochemical work of your muscles. So we will try and understand how this works energetically. Right? Now the production and utilization of bioenergy, what it requires is chemical reactions. And chemical reactions usually in a series of reactions, which brings us to pathways. So metabolic pathways here, what this really means is these are series of chemical reactions. And reactions that convert one compound, a compound being a metabolite, so a small molecule, into another. And for most of the quarter, what we'll be looking at are enzyme-mediated reactions. Sometimes these reactions can also occur spontaneously, but for the most part, what we'll be looking at and what is occurring in metabolism are enzyme-mediated, enzyme-catalyzed reactions. And then I already alluded to it, this name here, metabolites. Again, here, metabolites now, or chemical compounds, are low molecular weight compounds. So what we're talking about is small molecules, usually with a molecular weight of under 1,000 grams per mole. Right? So most of these are small chemicals that we'll be talking I mentioned earlier biomolecules. This encompasses a bit more. So this would include metabolites, but also larger kind of molecules such as proteins and so forth. In our case, mostly we'll be looking at small chemicals. So when I mention metabolites throughout the quarter, this is usually what I'm referring to, these low molecular weight chemical compounds. And so these can have different functions in metabolism, right? If we connect now the metabolites with our metabolic pathways here, how do they relate? If we think about an enzyme function, right? And we think about that enzyme now catalyzing the conversion of one compound into another, right? We have our one compound here. We would call that the substrate for our enzyme that is mediating this conversion and will convert the substrate into a product. Both substrate and product are metabolites. Okay? Obviously, metabolism is not just comprised of one enzyme. As I said, we have metabolic pathways, so these series of reactions. So it might go further, right? So this product here now might be converted by yet another enzyme into a different kind of product. So now what was a product before here, this first enzyme becomes a substrate for the second. And so this product now we can call a substrate, and oftentimes within the series of reactions within a metabolic pathway, we would call it an intermediate. Okay. So this can get rather complex. Just keep in mind here that metabolites can be substrates, can be products. Such products can be the substrate for another enzyme, then called intermediates. So they're different functions of the metabolites, and it's very dynamic in metabolism. Speaking then of metabolism, right, now that we understand what pathways are defined as and what metabolites are defined as, what metabolism is essentially is the sum of all of these pathways. Okay. So the entire sort of network of different pathways that are interconnected with each other, that we would call metabolism. But keep in mind, metabolism is not all the same. Okay. Metabolism always has to be viewed in the specific context. What I mean by that is that the sum of all pathways can be all pathways within me or a different context, right? So we're looking at basically all the reactions that are important to maintain the living state of an organism, like me, but it can also be the metabolism of a specific organ. So for example, the metabolism of the liver is different from the metabolism of the heart or the metabolism of the kidney. So it's an organ specific, right? It's in the context of a specific organ. We can go even smaller, right? Different cells have different metabolic states, a different metabolism, right? Cells in your brain have a different metabolism from cells in your liver again, for example. And in addition to that, we also need to take into account different environmental conditions, for example. You will have a different metabolism as you age. There are gender differences. Women and men generally have differences in their metabolism. Environmental impacts, also not just in humans, but in all other types of organisms and across the kingdom of life. So what I try to really hone in here on is that it's really important to try and view metabolism not always as a whole that is not changing, but a very dynamic network of reactions that have to be seen within their specific context. Right? That's something that I really want you to pay attention to as you try and understand biochemistry and metabolism. Then if we look one further here, metabolome, this might be a term that you haven't heard yet. So metabolome, as metabolism is the sum of all of these pathways and reactions. As metabolome, we would refer to the sum of all the metabolites. And again, much like metabolism, it is seen in the context. So it can be the metabolome of an entire organism, or it can be the metabolome of a specific organ, a specific current environmental condition, and so forth. Okay. One more term that I want to draw your attention to that you also may not have heard yet is metabolomics. And so this is a more recent term, and that really refers to a technology and specifically the high throughput analysis of metabolomes and metabolites. We won't go too much into detail in how this works this quarter, but it's something to draw your attention to because it's really one of the state-of-the-art technologies that science is using today to analyze metabolism. And so this is sort of the landscape in a simplified version. I obviously don't expect you to learn all of this. What I want to highlight here to you is how complex and interconnected metabolic pathways are. So what you see here, all these different dots are individual metabolites, substrates, products, and intermediates of different pathways that are shown here as lines. And you can see there are many of them. And you can also see that all of them are really heavily interconnected with each other. So while your textbook will illustrate it often as sort of an individual linear pathway, and we'll go this quarter through a lot of these pathways in many ways, just looking at this one pathway, please keep in mind that they're heavily interconnected and interdependent of each other. That's important to note. Just as a fun fact to sort of think about how many metabolites are there, usually I do this sort of as a little bit of an auction in class, but remotely here, I just want to draw your attention to how many metabolites are there in different organisms. So for example, if we look at E. coli, a fairly simple organism, a microbe, roughly 3,000 metabolites that E. coli produces and maintains for its own life. Right? If we go a little bit more complex, we look at a human being, we produce and maintain about 38,000 metabolites, plus minus, obviously, depending on the context again, okay? So quite a bit more, as you would expect from a more complex organism. But then what I find personally very intriguing is if you look at plants, which you may consider a less complex organism than a human, plants actually produce more than 250,000 metabolites. And I would argue that given how little we know about plant metabolites still, that this number is largely much, much higher. And so you may ask why? Why is an organism such as a plant that is presumably less complex than a human organism, why does it have so many more metabolites? And the answer here really is it's a different way of life. Right? Plants are sessile, meaning they are, for the most part, stuck in the place where they grow. So they have to have different ways of interacting with the environment. For example, in a situation of danger, you have a nervous system. Humans have a nervous system. You can use that to react to the environment and you can move away from danger. Plants cannot do this. So they use chemical means of defending themselves. They use chemicals to interact and communicate with the environment. And so then it makes much more sense and is more understandable that plants produce so many more metabolites because it's really their primary form of communication with the environment and interactions with the environment. So that's just for a fun fact. All right, so let's start diving in to metabolism, right? We just looked at how dynamic metabolism is, how interconnected these different pathways are, but we can sort of to start to try and understand metabolism, break it down into sort of some major differences in major functions. One of those differences is to divide metabolic pathways up between anabolic pathways that are involved in anabolism and catabolic pathways here. So these are two opposing functions, right? Anabolic pathways here are pathways that build molecules. So what they do is they take basically simpler metabolites such as amino acids, sugars, and so forth, and they will convert them into more complex metabolites. So just to give you one example, right, a simpler metabolite here could be an amino acid, and an anabolic pathway would convert these amino acids, for example, into proteins, much more complex, not metabolites, biomolecules in this case, macromolecules that are really much more complex. Okay. On the opposing side now, we have the catabolic pathways, right, where you have these complex metabolites such as polymers of carbohydrates, fats, or again, using this example of proteins that you, for example, get through your food, primarily in our case. Okay. And so for catabolic pathways, these complex metabolites now are being broken down into much less complex metabolites. And the purpose here is to use these breakdown reactions to generate energy. Okay. So while these two pathway concepts are opposing to each other, they are actually interconnected through a few specific metabolites here. And we'll go into much more detail into these throughout the quarter. And I like to consider them as sort of a common currency. Right? And specifically here, we have ATP, adenosine triphosphate, and NADH, a nucleotide dihydrate. And so these compounds here, they are sort of the common currency between these different opposing pathways, right? And this makes sense in many ways. I think a good example would be to think about our need, right? If we were to trade, like for example, if I would be breaking something down and I would want a pear, but you only have an apple, I can't really do anything with it, right? But if we exchange cash, a common currency, right, then I can take that cash that you got instead of the apple and I can invest it into my pear. So what I'm trying to say is that really having just a few metabolites here that are at the centerpiece between these two opposing pathway concepts really helps to have them more easily interconnected as a common chemical currency. Right? And at the same time, these compounds here, ATP and NADH, they also store our chemical energy, right? So the energy released from breaking down these complex metabolites into simpler ones, this energy will be stored in these molecules essentially now as a currency, as cash, that you can use this energy now to build up sort of these simple molecules here into more complex ones. So the energy needed for this is coming from your common currency here in form of ATP, NADH, NADPH, and FADH. Again, the details on all these molecules will go throughout the quarter. So this is just to show you sort of the broader concept of what we'll be looking at. All right. So some more terminology to get us started. We had started to talk about catabolic pathways and anabolic pathways. So again, written out here, I like to keep in mind catabolic pathways, their major purpose is energy releasing. We're breaking down complex compounds into simpler ones to release the energy that is stored in these complex molecules. The energy primarily is captured in ATP and adenosine triphosphate. And we'll look at how this works in really a lot of detail. In addition to the energy, we will be using a lot of oxidation reactions in these catabolic pathways, and these oxidation reactions will release electrons. Again, these are also containing energy in a different way than we will see for ATP, but these electrons are captured in NADH and FADH and can also be used later for our anabolic pathways, right? So moving to those, the anabolic pathways, then they will take simple compounds and convert them into complex compounds, and they will use the energy it is stored in ATP and NADH primarily. Right? And they will also, what these pathways do is they will provide biosynthetic precursors. Right? We'll look into this. So they will provide sort of intermediate complexity kind of metabolites that we need to make more complex ones. And I just want to highlight here, there are some pathways that we call amphibolic. So these are pathways that can be functioning both as energy building and energy releasing pathways. There are actually a few of those, and we'll highlight a few examples. But primarily for this quarter, what I want you to focus on are these opposing catabolic and anabolic pathways. Okay. Something that are common among these in terms of properties down here on the slide, what I would want to draw your attention to is, again, all these pathways occur in steps, right? This is very important, and they're typically enzyme catalyzed reactions. That will be our focus by far. One thing very important, catabolic and anabolic pathways can share steps. So while they're opposing, some of these chemical reactions can be shared, but really not all. This is really important, and I want you to remember that and pay attention to, they can share steps, but they cannot share all the steps. And we'll be looking later on in the quarter on why that is. Okay. And then again, as I mentioned before, they are linked through the common currency ATP and NADH and NADPH in some cases. All right, so much for terminology. Let's look at what we actually want to cover this quarter. So I have listed a few sort of highlights of topics that we will go through. You've seen how complex metabolism is. We will only focus on some of the major pathways that are really critical in this context and that have sort of the largest flux in metabolism. So the largest, essentially, rate of metabolism going through these pathways as flux. This also will largely cover sort of the essential and conserved pathways, and we'll look at these conservations across different organisms. We'll focus on microbes and humans primarily, but also look at some shared and unique pathways that we find in plants. Right? And so what we'll do is that we'll go through these concepts and just choose a few example pathways that we can go through, and we'll use those examples to try and understand pathway regulation. Like how do these pathways work with each other? How we can use these pathways to sustain life under all the different circumstances that we encounter. Okay. And then some of the major metabolite classes that we want to look at this year are our sugars here, the carbohydrates. We look at proteins and amino acids, and we look at our fats and lipids this quarter. So these are sort of the big three as they relate to energy metabolism and many of the really critical functions in metabolism that we want to focus on. All right, so this will be our focus. Today, again, I just want to give you a bit of an introduction, and this also would include how can we study this? Just to give you some ideas on how the knowledge that we will discuss this quarter was actually generated using experimental research. And so this, over the years now, has become really complex. In order to study metabolism, we were drawing on a multitude of different technologies and experimental approaches that are now at our disposal to try and understand this complexity of metabolism. Right? And so this includes currently a lot of what we call omics technologies. So these are genomics, transcriptomics. I already mentioned metabolomics and metabolome to you. These are high throughput tools to generate large sets of data on metabolites, genes, and enzymes that we can utilize. And we can combine these with more specific experiments, such as biochemical experiments, for example, enzyme functional assays, those types of approaches. We can do genetic experiments, so we can actually modify and understand the function of a gene in its natural environment. And we can do so by trying to understand natural variation. So for example, right, we can try and understand metabolism across a variation of humans at different ages, for example, or different genders, different demographics, to try and get a more complete understanding of what is happening. And all of this, and we obviously want to put into the ecological context of biological interactions, evolution, climate, all these things, to try and understand how metabolism functions in the context of life in itself. Some sort of specific techniques that I want to highlight to you and that we will look at this quarter more frequently are in vivo techniques, so in the living organisms. There are things like isotope labeling in living organisms. I'll show an example of that. We can use inhibitors to block pathways, and that's a really helpful tool to try and understand these series of reactions. Instead of inhibitors, we can also use genetic mutants, and there are many, many tools at our disposal today. Again, the same purpose here, to block or overexpress pathways to understand these series of reactions. And then outside of the living system, so in vitro techniques, there are many of those. I'll just highlight a few here. Subcellular fractionation has been used for centuries to try and understand metabolites and their functions. So there you would use, for example, extracts from different human tissues and try and understand what metabolites are in there and what their function might be. We can be more precise than this, and we can isolate individual enzymes. We can characterize their functions. And again, we can use also in vitro these many high-throughput omics type techniques to try and answer questions about metabolism. Here's one example. This is an example of isotope labeling. So this is a very early experiment from about the 1940s that was used to try and understand photosynthesis. And what you see here are radio-labeled metabolites. So what they have done is that they fed radio-labeled carbon dioxide to an algae. And then what they have done is they have monitored the development of different radio-labeled metabolites here on a thin layer chromatography. So the principle is that you feed in this radio-labeled carbon dioxide, the metabolic pathways occur, and they will incorporate the carbon dioxide into the products of each of these enzymes. Okay, so the first enzyme will take carbon dioxide, will catalyze a conversion of, again, a simpler metabolite into a more complex one that now contains this radio-labeled carbon dioxide. Therefore, the product of this enzyme will show up here as a radio-labeled metabolite. Right? And now you can just follow the series of events, the series of reactions in this pathway. Okay. And I want just to highlight here, right, you see the time stamp here. It's 5 seconds. So this is an experiment they have done. And after 5 seconds of feeding radio-labeled carbon dioxide to this chlorella algae, you already have a whole number here of different intermediates occurring. There are sugar phosphates here. There are already acids being produced. And even small amounts of amino acids have already been produced. Right? So you can see how quickly metabolism occurs. Within a few seconds, you already have moved from incorporating radio-labeled carbon dioxide to having produced an amino acid. That's really fascinating to think about how efficient metabolism actually is in terms of its timeline. This is an example of using inhibitors. So these, for example, can be chemicals that block an enzyme function in a metabolic pathways. Okay. So on the left side here under A, what you see is, right, a metabolic pathway. You have a substrate, a number of 6 enzymes here that will go to an eventual end product of this pathway. And if this pathway is undisturbed, you should be seeing sort of regular amounts of all the intermediates here, B to E, and the final product of this pathway here, F, to occur. Okay. So now if you use inhibitors, you can ask questions about what are these intermediates? And if you know the intermediates, what might be the enzyme functions in this pathway? Okay. So how can we use inhibitors for that? So now assume you have the same pathway now here on the right under B, and you use a chemical inhibitor. So an inhibitor that will now prevent enzyme 4 to mediate or catalyze its function. Okay. If that happens, right, theoretically now we cannot produce E anymore, we cannot produce F anymore. And this is reflected here in this bar graph of the abundance of these different metabolites. And you can see indeed, if enzyme 4 is blocked, we have much less E and F produced. Right? So this might be an inhibitor that is not fully inhibiting, fully blocking this function. You still have a little bit of this happening, but it's largely reduced. And instead, the substrate of the blocked enzyme 4 now accumulates. Okay. And so now if you can analyze this metabolite and identify what it is, right, you can infer from this metabolite what the function of enzyme 4 is. And so you can use these inhibitors now just by walking through this series, right? You do one for enzyme 3, enzyme 2, and so forth. You can develop knowledge about this entire pathway using this fairly simple type of experiment. Okay. Like I said, instead of inhibitors, you can also use the modification of genes, so blocking gene functions genetically these days. Many of you may have heard about CRISPR. So this is a technology that essentially can create cuts in DNA, and it can allow to edit a gene. And so if you think about this, right, if I cut in this gene, I'm making modifications to this gene. I can, for example, cut out a certain piece, or I can put it out of frame by adding additional nucleotides. The result will be that this gene and the enzyme that is encoded by this gene will not be functional anymore. Okay. It's a genetic way of doing this. The principle is very different, but the outcome is the same, that now, much like with an inhibitor, the function of the encoded enzyme is blocked. The same principle occurs, that you now can see accumulating metabolites as well as those that are not accumulating anymore, and can make deductions on what these genes and enzymes may have as a function and what the metabolite is, and therefore understand metabolic pathways. Then are here these omics tools that I mentioned. So these are high throughput tools of doing this. So for example, metabolics now could be used if you think about doing a series of inhibitor studies or genetic modifications. These omics tools allow you to analyze a large number, thousands of metabolites in a very short time, to really understand what is happening in metabolism when I use this inhibitor, if I use a modified gene. Okay. So metabolomics is this high throughput technology to analyze metabolites in an entire organism, a tissue, a cell, depending on the context you want to ask your question in. Okay. We can do the same for proteins. This is then called proteomics, very similar kind of machinery, but focusing on larger molecules, proteins in this case, and peptides. And then genomics and transcriptomics is the same high throughput technology idea, but it's designed to understand genomes and transcriptomes respectively. Okay. This is a different type of analysis. These are sequencing machines. And so you may have encountered them if any of you have done so. So 23andMe, this is exactly the technology they're using to sequence your personal genome. They're using these genomics and transcriptomics types approaches using high throughput sequencing machines. So these are just some of the sort of broad tools. We go through some of these tools in more detail as we're encountering individual pathways. But to give you an overview of what is out there, what is available for studying metabolism. So the next big question is why? Why should you care? Why are you sitting in BIS103 and watching this lecture? Why is metabolism important? And I thought I'd just highlight sort of a few examples of what I think are sort of the key factors of why this is important. And so why do we care? It's actually been a long time, a long-standing interest of humans. So there are actually some studies that have shown that even 60 to 30,000 years BC, humans have used medicinal plants. Okay? So there have been some studies done in the teeth of mummies, and they could identify that what they have eaten are things like wormwood or yarrow. These are medicinal plants, and they could be identified based on the metabolites that were extracted from the plant material in the teeth of these mummies. And so our interest in metabolism for health purposes has been really long going. And then through the centuries, we continued this interest, right? Throughout the centuries, there have been more and more work on trying to understand what the metabolites are. Initially, a lot of work done in plants. Regrettably, we had a big interest in developing poisons like conein here, developed from Conium maculatum. It's a famous metabolite that, for example, was used to execute Socrates, as legend tells us. There are some more beneficial compounds like caffeine, that's an alkaloid metabolite from the coffee plant. Most likely you all know that. Atropine here is an example that legend tells us Cleopatra used. It's actually a compound that will widen the pupils, make you look more appealing. It's actually also used for eye surgery. Right? It's still actually used as a current drug. And there are some more infamous siblings of those. Nicotine, for example, is what you would find in tobacco. But this biochemistry, the knowledge of metabolism of these plants has led to all of these developments and discoveries. And then more recently, right, we've understood from the large historic knowledge we have, if you think about Chinese traditional medicine, if you think about Ayurveda, we have a wealth of knowledge of plant natural products, plant-derived metabolites, as well as from other organisms that are really useful for the development of treatments and of drugs. I just want to highlight insulin here, one of the major important drugs to treat diabetes. And insulin was discovered early on. You can see this package here is from 1923, and it was discovered by using this approach I mentioned earlier of fractionation. So what people would do is they would use, at that time, the pancreas of dogs, and they would fractionate the pancreas tissues through different approaches and just test which of these fractions would have activity and would keep the humans or the dogs that they would feed with these fractions healthy. And so eventually this was what they used to identify the function of insulin, which is really one of our major drugs today. Of maybe even more importance or equal importance is the development of antibiotics, right? Sort of going all the way back. Some of you may remember Alexander Fleming, who developed penicillins, as legend tells us, a bit by accident, by being sloppy and having this fungus grow on a plate. But it was the origin of our work on using natural resources, especially fungi and plants, to develop antibiotics. And you can see throughout the decades since the 1920s, we have developed numerous different antibiotics. But in recent years, there has been a real void of the development of new antibiotics. And you're probably aware that we have sort of this big issue of increasing resistance of especially microbial diseases against our current antibiotics. And we are in urgent need of developing new antibiotics. And so there's a lot of literature. I'll just highlight one example here of research that is being done to try and find new, more potent antibiotics. And I would argue it's very timely right now in our COVID-19 crisis. Antibiotics won't help here. It's a viral disease, not a bacterial disease. But you're certainly aware from the news how active research is at this time to try and understand the metabolism of the virus and to find a vaccine that we can use to treat it. Then we can also use the understanding of metabolism to produce these compounds, not just the discovery of them, right? We can use our understanding of microbes here and of their metabolism to actually introduce different enzymes and then make these microbes essentially produce chemicals of interest. And so this is called synthetic biology or metabolic engineering, where once we know the function of, say, an enzyme from a plant that produces a metabolite that can be a drug, a therapeutic, we can then use our understanding of the metabolism of this microbe, introduce this gene in the microbe so it produces our enzyme of interest. And so then we can extract, right? The enzyme will be active in the microbe, it will produce a metabolite, and we can use this for a number of things, right? In this case here, this is actually the facility of Ameris. So what you see here are huge fermenters. There are 200,000 liter fermenters growing these microbes. And what they're producing is artemisinin, which is a metabolite from a plant that is actually used as a drug to treat malaria. So all of our metabolic knowledge we can actually use for industrial purposes beyond discovery. And it's really a crucial part for a lot of industrial production of bioproducts, all the way from fuels to drugs, flavors, fragrances, other fine chemicals, and so forth. And we'll see a few examples as we move through the course. And last but not least, that's very close to my work, we can understand and use understanding of metabolism also for improving our food, right? If you can imagine, right, the more we understand, for example, how metabolism, in this case here, of a corn plant changes, if it is attacked by a microbial pathogen or an insect, for example, we can use this knowledge of metabolism to improve the resistance of this crop. So here you, for example, see now corn lines that have been bred toward changed metabolism that now is more effective in fighting off this pathogen. So here you see all the necrosis that is a result of the pathogen. And here you see really nice, healthy plants. And so these developments really have been built on understanding the metabolism of the plant in the context of being attacked by a pathogen. These are the examples I have for today. So this is just meant to be an introduction. We'll start to dive in deeper into our first pathways in the next lecture. So I'm looking forward to seeing you there.